Right, so yeah, I want to talk today about balancing depth, throughput, and pragmatism in plasma proteomics, because as most of you will know, plasma proteomics is not very easy to do. And before I do that, I just want to quickly introduce basically the lab I'm working in. So this is the Discovery Proteomics facility at the Target Discovery Institute in Oxford. We are a small research lab doing lots of service work, but also primary research on proteomics. And the work I'm showing today here has mostly been done by Raphael and Georgina in the lab. So when it comes to plasma proteomics, I think most of you have already seen this so-called Anderson ladder here, which basically shows the dynamic concentration range of proteins in plasma or serum. And as you can see here, we have here a logarithmic scale. That means basically that the bulk of the protein mass in plasma and serum is made out of a very small number of proteins. So if we looking at this here, we have about 10 to 11 orders of magnitude in dynamic range in the proteome of plasma, which makes it a hugely difficult analyte to look at by basically detectors that can, on a good day, cover, let's say, 4 to 5 orders of magnitude in dynamic range. So basically, if we have a sample that contains all these proteins with a standard LC-MS platform, we might be able to get down to here, to these kind of small number of proteins that will be in the low hundreds, maybe even only 100 or 200 proteins here that we can actually see in these types of samples. So the dynamic range of the proteome itself is one of the problems. Another one is that the proteins themselves are also abundant in a vast or really high dynamic range between, for example, patient samples. So here we have one of the most known biomarkers, PSA, which you can see here spans a concentration range of like 4 or 5 orders or more orders of magnitude in dynamic range. The same is true for other known biomarkers, for example, like CRP, where we easily spend 3 orders of magnitude from baseline as compared to clinical condition, like some autoimmune diseases. So that is a second basically big problem in plasma proteomics that we have facing here. On top, we have other things that basically sort of impede easy sample processing, like high lipid context or the generally high protein concentration, which can lead to problems when you sort of freeze store your samples and things start to precipitate, which also is for sample handling a big problem. So when we want to do plasma proteomics, there are some solutions to address these problems. When we look at dynamic range, the most commonly used ones are depletion. So these are basically little, very often spin cartridges. You apply your plasma or serum to them, and they kind of have antibodies that remove the most abundant, for example, top 2 protein species from the plasma, top 12, top 14, and so on. And that what you gain basically by doing that is this like 1 or 2 orders of magnitude in the top here, so by removing those. But even if you're here now, for example, after applying a top 10 column, again, don't forget you only still can detect like 5 orders of magnitude in your mass spectrometer. So that doesn't really help you a lot by going further down in the plasma proteome. And this is basically where this approach comes into play. So here we have a tandem depletion, where we first remove the most abundant 14 proteins in the plasma or serum, which is then followed by a second antibody-based column that removes the most abundant 50 proteins subsequently in plasma. And by doing so, then we suddenly gain like 4 or 5 orders of magnitude in dynamic range, and that then allows us to go much deeper into the proteome. Unfortunately, all of these are really quite expensive. So these are antibody-based columns, and a set of these 2 columns that you can purchase commercially sets you back about £20,000, and that then is sufficient to analyze 100, maybe 200 samples that you get out of it. There are other depletion technologies or methods. What I like in particular is the biochemical depletion, where we use very cheap and cheerful protein A and protein G beads to remove IgGs from the serum or plasma, and then do like a biochemical depletion using ethanol, where basically everything precipitates but albumin and stuff that is bound to albumin and other more globular proteins. That is very cheap, can also be done in semi-high throughput and then is actually one of the pragmatic solutions to deal with the dynamic range of the proteome in plasma. These are just some comparisons, and I don't have time really today to show you a lot of data, but this is comparing a single shot analysis of these top 14, top 12, and top 64 depleted plasma serum samples. And here we used a commercially available serum that you can buy of Sigma, which will not contain basically the more sort of interesting proteins to high levels. So we always are expecting to detect lower protein numbers in these commercial serums. So we have here basically the top 12 and top 14 depletion methods, which perform more or less the same. If anything, top 12 maybe is a little bit better when you look at the number of unique proteins that we see here as compared to top 14. But we gain basically by a single shot analysis the most by using top 64 depletion, so these tandem depletion columns, which are really expensive, where we gain like 200 exclusive proteins out of there's some like 700 or 800 proteins within the whole experiment. When we do a comparison between a non-depleted serum in this case versus the top 12 depleted serum, we can see that actually we can get a good coverage already without depletion, and this is quite a constant number of proteins that we can detect in each of these runs, and it kind of is independent on the gradient length more or less, and the sort of sophistication of the mass spectrometer attached to it. So even a like 5 or 10-year-old mass spectrometer is able to detect as many proteins with the same amount of material. So, but what I think is remarkable here, and this is actually not depicted very well here, is that if we would just use higher numbers of samples, so if we would go up to like 20, 30, 40 samples, we would actually eventually reach the same number of protein IDs as in the top 12 depleted sample. So that means that ultimately, when we have high N numbers, we will reach a similar number of identified proteins. However, we will reach that plateau much later as compared to these top 12 depleted or depleted samples in general. And of course, we will have more missing values in these non-depleted samples. So here's one of these extremes, and this is the top 64 depleted sample, and this is basically driving it much further to maximize protein identifications. So as I mentioned, we have a column which contains 50 antibodies against basically these 50 proteins that are very abundant in plasma, and everything that is not these 50 proteins will basically elute with the void volume of the column when you hook it up to an HPLC or FPLC in this case. So that means basically what we are after are these proteins that are not binding to the column. There's not really much chromatographic separation going on here because it's in the void volume, but we can still fractionate these, and we will have different proteins in these different fractions. So basically we can, for example, get these 10 fractions as shown here, and we can then digest these fractions individually and do a high pH peptide level fractionation on top of that into 15 fractions. So we would end up with 150 samples, which can then be quite quickly analyzed by an Evosep timsTOF Pro in this case platform with 60 samples per day. So we would spend a little bit more than 2 days on one of these blood plasma samples, and then we actually can get 3,500 or so proteins identified in these samples. So by basically effectively removing these proteins spanning these sort of top orders of magnitude here, we then can basically start detecting, and this has not changed. We still are bound to our like 5 orders of magnitude of linear dynamic range in your mass spectrometer. So basically we can go and clock down to here roughly, where we actually start to see chemokines and cytokines appearing in the data, and also get a lot of tissue leakage proteins covered. So this is really quite nice. However, it's expensive. I also want to raise a little point about quantitation. I think this is well known, but I just want to show some data addressing this point, is that any kind of depletion will affect your quantitation. And this can best be compared to a sample that is not depleted, because that is basically the natural state of your serum or plasma, where we have not fiddled with really high abundant proteins. So this we can see as a reference. So if we just take 2 replicate preparations here of non-depleted serum samples, we can see that we can get very nice agreement between these 2 samples, very good correlation factors. But the moment we actually fiddle with highly abundant proteins, we will have an impact on the proteins that we can detect and also how we quantitate them. So if we make this comparison between non-depleted sample versus IgG depleted sample, and this is again injecting the same amount on the same system, we can see that our correlation between the two different methods sort of degrades very quickly. So we only get like a 0.8 or 80% correlation between the two. This gets worse if we deplete more abundant samples. I think we can kind of explain these proteins on this end here. These are the proteins that we have basically almost removed from the plasma, that have very low intensities or non-existing intensities in the top 12 depleted sample, but still have significant intensities in the non-depleted sample. But still you can see that we have introduced a lot of variance also for everything that we are not actually touching, stuff that we are not depleting. So, and that comes from, for example, that we are removing proteins that are carrier proteins. Everything that sticks to albumin, for example, is also being removed in these depleted samples, and that has a severe impact on how we quantitate. This is also not really better if we remove even more. So if we remove top 64, we still get a very bad correlation. And we can see that and understand that whenever we wanted now to go ahead and do an absolute quantitation approach, where we would spike heavy peptides, for example, into a depleted sample, this doesn't really work anymore. So the numbers we would sort of infer in terms of absolute protein concentrations for these spiked peptides basically have nothing to do with the real concentration anymore in the actually depleted samples. So we can only do this on not depleted samples. Okay, and basically after all this is basically where we were when the global pandemic hit, and this kind of changed quite a lot of things very quickly and changed perspectives. And also, if I had, or if my younger self would also look at me right now and be a bit surprised on what I'm sort of advertising in terms of how proteomics can be done. And this is kind of summarized in this little picture here. And we have to be pragmatic when we do plasma proteomics, especially in large clinical cohorts. And the first thing I want to mention in this context is the N number, because this very often is still underestimated. Basically, how many samples do we actually need for a significant or relevant comparison between disease states? So, and this plot here nicely shows that if we have, or if we assume that we have high variability between clinical specimen and patients, and this is really true, so we can easily get something between 75 and 100% overall variation between patients or plasma samples from patients, and we want to be sensitive enough to detect like a 1.5-fold change with a power of 80%, we need something between 30 and 60 samples per group. So this is relatively straightforward and still doable with low throughput if we have just a simple comparison, disease versus control, that would then still be doable and feasible with low throughput platforms. But the moment we add covariants like age and sex, then we suddenly have to multiply the sort of number or N number that we have sort of come up with the number of groups that we are looking at, and then we are looking at suddenly hundreds or even thousands of samples. And this was the case the moment the pandemic hit, because we were sort of asked for action to be very quick in analyzing lots of COVID patient samples. So, and this is where we had to become pragmatic. So I mentioned we have to deal with large sample numbers. As I mentioned, we don't even know what the groupings are. So we don't know what kind of age groups we are going to define, where at that moment, at the beginning of the pandemic, there was not really clarity about where kind of the age thresholds are, where symptoms would get very badly or very bad. We didn't have consistent sampling methods. So there was a lot of stress in the clinic. The samples were not really prepared and accumulated or collected in a consistent way. We didn't even know the grouping, as I mentioned at that point, and we had pathogenic samples with a pathogen where there's neither treatment nor vaccine. So we had to be very conscious of these samples being potentially dangerous. That is not unusual. However, I think this was still a very special situation with these pandemic samples. So what this required was that we had to immediately inactivate the virus. There was very little known at that point about how to do this. There were some preliminary studies where we could use IPA to inactivate the virus, and this is ultimately what we had done, and that basically precipitates the proteins right out of the sample. So there was no way of doing any kind of depletion or even doing sophisticated proteomics sample preparation as you would expect using DDT and uracilumide, for example, to alkylate your cysteine. So that was all not really feasible. We also needed high throughput sample preparation. In this case, we would not go into our liquid handling platform, we would actually use multi-channel pipettes with many people. And then we had to employ high throughput sample analysis because everybody was very eager to see the results, and also high throughput data analysis, which at that point was not really feasible with large numbers of samples. And after all this, we had kind of generated our data. We needed a lot of patience because it turned out that all the other omics technologies that in our consortium were applied to the same number of samples basically took a lot longer than proteomics. And this was quite a new experience for us because usually proteomics is one of the slower technologies to generate the data. Yeah, and I just want to highlight here the COMBAT consortium, and this is basically lots of people, mostly sort of being in Oxford in multiple different institutes that all contributed to this consortium to analyze the same set of samples with a maximum of different omics technologies. And then we just provided the plasma proteomics data for this. So we basically took all the samples that were available in this cohort, and these turned out later to be healthcare workers, which were basically COVID-19 positive people who were not hospitalized and had very mild or no symptoms. And then we had hospitalized cases with mild, severe, and critical symptoms. We had some sepsis samples to compare, and also some healthy samples as well. So some heroic efforts from these guys here in the lab who sort of went through all these kind of preparations and prepared these samples in a very efficient and quick way on the weekends, long working hours, and so on. And then we generated the data, which was then subsequently analyzed in FragPipe. And here, big shout out to Feng Chao and Alexei in the FragPipe team, who helped us basically by developing or further developing their software on the fly to cope with the number of samples that we had. So we had ultimately almost 600 mass spec runs here in this cohort. And we actually, on non-depleted samples, we actually identified a lot of proteins here, 702 in the whole data set. But it turned out that in each individual sample, we did not identify so many. So we got something like 160 proteins identified in each sample. And this is what I meant, that we reached this plateau of a maximum, in this case of 702 samples, very late with non-depleted samples. With depleted samples, we would reach this much earlier. Ultimately, we got only like 83 samples for which we had like intensities, 83 proteins for which we could get intensities in each of these samples. So that led basically to these complicated plots. We have here correlation analyses. On the left one, we have a supervised version, which shows basically that when we look at the, in this case, healthcare workers, for example, in yellow, they correlate really well with each other and not so much with the more severe disease cases. And this is using 107 proteins basically as a matrix to create these correlation analyses. If we do an unsupervised clustering, we can see that basically the patient samples assemble themselves really nicely together within their patient groups. For example, here we again have the non-severe COVID cases, non-hospitalized COVID cases right next to the healthy controls. And the further we go to the right, we get into the more severe COVID area where we have significant or, well, specific protein profiles that sort of highlight and correlate with the disease states. This is also obvious when we look at principal component analysis of all these data, and you can see that here the healthy controls really nicely overlap with the mild or non-symptom-free corona cases. And while we go further to the left alongside principal component 1, basically the disease gets more severe, and then we get some overlap with sepsis as well. And this was done together with Phil and Yoshin, and as well Alberto, using also the clinical knowledge graph that he has developed. So we employed in the consortium all these different omics technologies. So with proteomics, and the timsTOF. This is just this line here, but there were lots of other things that were done on the same set of samples. So we basically generated a huge multi-omic data set, for which in this kind of scale, there's actually no way of, or no sort of golden rule how to integrate the data. We made some attempts, and we were very pleased to see that when it comes to patient disease severity stratification, our mass spec data actually provided the best data in the field. And you can see that basically principal component 1 and 2 together basically are sufficient to stratify patients according to disease severity. When we look at the loadings in these principal components, we found that these two proteins, SAA2 and CRP, are the most informative. There are some other proteins that have been provided by Luminex analysis, which basically also still not able to stratify the patients to the same extent as the proteomics data alone can do, which is really nice to see. When you go on medRxiv and search for COVID blood atlas, you will see the paper. It's a vast amount of work, a huge paper that is still in review. I think it will take quite some time to review this data. And this is really just one little output, one example, and you can really go and look at the paper for much more of this, and basically about how we try to integrate the data. And the first step here was really just to assemble matrices or tensors, multidimensional matrices that kind of ignore the modality, but put all the data that basically show a certain sort of abundance profile in this case, and here we have basically nothing in the, or very little signal in the healthy controls, increasing signal with disease severity, and some signal in the sepsis cohorts, and not much signal in the community COVID cases. This profile is being used to cluster the data, and we can see that certain monocyte populations basically cluster with this. We have a CyTOF analysis or sort of proteins that cluster with this type of profile. We can look at gene expression profiles and find some that will cluster with this profile, and so on, and ultimately also some of our proteins that cluster with this, basically. Right, just a little perspective. I mentioned that we have to be pragmatic when we deal with large sample cohorts in clinical proteomics and plasma analysis. This is even more important if the cohorts get bigger, and we are facing bottlenecks that we have not experienced, at least in the proteomics field, so much before. So this is just one example where we had 3 sets of these kind of 10 boxes each of samples, so 2,600 samples in total. And you can imagine that even just to get these samples into a format that we can actually use, for example, using a liquid handling robot, already is a task that can take weeks to achieve. And at the moment, we are experiencing these bottlenecks. And then if you then think about adding complicated sample preparation, plasma depletion, and things like that, this is all way too complicated and something that is not really feasible at this scale. So we have to be pragmatic and employ a really simple sample preparation workflow, which is then followed by a high throughput proteomics approach. And also, and this is also key, a high throughput data analysis approach, which cannot take longer than the actual data generation to deal with these large cohorts. And with that, I'm at an end. And thank you for your attention. I'm sorry I ran a bit overboard, I guess.